Well, good morning. This is Miss Dagenford, and we're going to be looking at how molecules move across the membrane, or as I like to have fun saying, red rover, can they come over? And so this is dealing with the movement of molecules through the membrane to the cell. And so by the end of this movie, you should be able to describe diffusion, describe the types of solutions, and explain why some molecules can and why some molecules can't. So first things first, let's go ahead and clarify diffusion. And so diffusion is the movement of molecules from an area of high concentration. And concentration, you might recall from chemistry, is symbolized with the brackets. From an area of high concentration to an area of low concentration. And the typical, I'll leave that there for a second. So the typical picture is that of molecules going from high to low. Now, what we're going to add is a semi-permeable membrane. And a semi-permeable membrane simply means only some things can get through. But you can notice in this case, the yellow molecules, they're moving, and what they're eventually going to do is reach a status of equilibrium. And that simply means that we've got the same, they're evenly spread. And they're doing this because they're colliding into each other, and they're colliding into each other because they have an inherent kinetic energy. Now that means that this is a passive process. You don't need to make this happen. They have energy because they're molecules, they're moving. Now we can't influence this with temperature. If the temperature increases, then we're going to increase the kinetic energy, which does explain why hot coffee is easier to dissolve sugar, whereas cold iced tea, some of you may have seen with frustration that the sugar doesn't want to dissolve with that. Now, another thing to notice is that the molecules are moving along their own concentration gradient. Now, a concentration gradient is simply a difference. And so think about a hill, and if you put a ball on the top of the hill, it rolls down naturally. So the high part of the concentration would be the high top of the hill, and then the low part of the hill would be the low part of the concentration. So molecules move naturally down their concentration gradient. So in the orange would be high going to low, but then notice the purple is moving high to low according to its own concentration difference. So there's your short, sweet, to the point definition of diffusion. Now, the semi-permeable membrane gives some interesting twists to the tale. If you have a semi-permeable membrane, that means some things can't go through. If we look at a magnified picture, these green molecules are way too big. And so these green molecules, as they move, they might have a tendency to move in the less concentrated area, but they can't. They can't get through this. So in the left-hand picture, I'm just drawing the fact that these molecules are just bouncing off. They can't move into the less concentrated area. So notice we've got low concentration of green, but that means there's a high concentration of water. Conversely, we've got a high concentration of green on the right-hand side and a low concentration of water on the right-hand side. So think about the direction the molecules want to move. Green would tend to move to the left side, but it can't. Water would tend to move to the right side, and it can. So in a demonstration like this, water is going to move up the tube, and we actually could do this demo. It actually would try to do it. Now, in this case, it's never going to be able to reach equilibrium, but it's going to try. So that kind of gets us into some interesting things that are happening with the semi-permeable membrane. Now over here we have an example of the three different classifications of solutions. We have a hypotonic, an isotonic and a hypertonic. Now the isotonic is the easiest to know because iso means same. Think isomer, isotope, isosceles, that all refers to the same something. So this is the same concentration. So the same amount of solute on the outside and the same solution of water on the outside as the inside. So in all three pictures, notice I used the same. I used 2% solute, 98% water, 2% solute, 98% water, 2% solute, 98% water. So what I'm going to vary is the outside. In this case, because it's the same, we'd have 98% water, 2% solute. That's the same. In the case of a hypotonic, we have fewer solutes. So it is a lower concentration of solute, but then conversely, that means a higher concentration of water. So let's go ahead and put some numbers down. On the outside, I might have a 1% solute, but that means I have a 99% water concentration. So notice this ends up meaning there's a higher water concentration on the outside. That causes water to go in. And in the case of an animal cell, that means it bursts. In the case of a plant cell, the cell wall is there, so the plant just absorbs water and it gets really, really, really full. And we call that turgid. It's full of water. The vacuole is full of water as well. So a hypotonic solution causes cells to gain water. Now, some students like to think hypo like hippo, and that's okay. That's one way to remember, but that's a good way to just help yourself on test anxiety. You still need to understand it. So if you want to remember hippo means that they gain water, that's okay, but you still need to be able to apply it. So we need to make sure we understand it too. So we're going to do some practice problems. So we have a hypotonic solution, isotonic solution, and now let's do hypertonic. In a hypertonic solution, the hyper refers to more solutes on the outside and less water on the outside. And so let's go ahead and put some numbers down. A hypertonic solution would be something like an 8% solute solution on the outside, and that means it would be a 98% solution. I'm so sorry, can't do my math. That would be a 92% water solution on the outside. And so we'd have more water on the inside, less water on the outside. Water is going to leave. And so in a hypertonic solution, cells lose water. And then in an isotonic solution, water is entering and leaving at the same rate. So they neither gain nor lose. So in a plant cell, what happens is they do lose water. The cell wall can't help that. So we call this situation flaccid. And so what we see it as the plant is wilting. So we're going to do some practice problems, but let me ask you a question. Here's one practical application. In a paramecium, and the paramecium is in a freshwater. These are little protists. And so in a freshwater situation, this would be a hypotonic solution. So that means there's more and so you had to answer that question. So there's more water on the outside in a hypotonic, and there's less water on the inside. And so think about what's happening. That means the cell is going to gain water because the water is going to move in according to diffusion. Now that means the cell could potentially bust. The cell membranes could burst. And so what they have is this little organelle called a contractile vacuole. And so it'll eventually fill up with water. And when it fills up with water, it gets really big and full. And then the contractile vacuole has a little pore to the outside, and it squeezes and pushes the water out. and then it returns back to that shape. We'll watch a little video of it. It's a pretty cool thing. And so in that way, the paramecium can ingest the water. And I want you to be thinking of some other examples of organisms that have to contend with this. Ocean-dwelling fish and birds, they're constantly in a hypertonic solution. Animals that return to freshwater, like salmon, well, they were in an ocean, but then they return to freshwater to spawn. So how might they deal with things? And then just as a quick side note, we have little cilia. That's how they're moving. So these little blurry things that you're seeing, that's the cilia. Now, facilitated diffusion happens because the molecules are either big or polar. And so here's a situation where the molecules just need to pass through a transport protein. Now, these proteins, the simplest ones are just pores, where you have polar side chains on the inside of these, but then the non-polar sections are facing the tails, and they allow for a channel to go through. But they can get more complicated. You have voltage gated uh, uh, um, or a receptor that opens them up. And so just as before, we're going to have a diffusion. So we're going to go high to low. Now the key is size and polarity. So a little tiny molecule like this can get through. So what are the rules? Well, the rules are the smaller the molecule, the more nonpolar it is, the easier it is to get through. The larger the molecule, or the not more polar it is, the harder it is to go through. So I'm going to just leave you with some examples and have you see if you can sort it out. Oxygen is really small and nonpolar. Carbon dioxide is really small and nonpolar. And then glucose is really large comparatively, and it's also very polar. And sodium is small, but it's fully charged. So think about that, and we're going to answer some questions. Have a lovely day.